Pirates, today's topic is DNA replication. So this is going to go on page 43 in your interactive notebook. And what we're going to look at real quick is just how DNA replicates in a living thing. So the essential question here is, how does DNA replicate or make a copy of itself to conserve the genetic code? So we're just going to start with understanding some who, what, where, when, and why DNA replicates, and then we're going to look at the actual process. So starting with the who, all cells, so all living things are going to need to replicate themselves. And so when they do that, they're going to need to replicate their genetic code. So all living things go through DNA replication. So, this is going to happen in cells. What is DNA replication? It's making a copy of DNA. So this is making a copy of DNA. When does this happen? It happens before a cell divides. So before a cell divides to make another cell, the instructions are copied so that each new cell has a copy of the instructions for the cell. So before cell division. And in a couple weeks, we're going to learn that this is actually going to happen during something called S phase of interphase. So I'm just going to preload you with those words. So it happens during what we call S phase of interphase. Where does it happen? Well, in eukaryotic cells, it happens in the nucleus. If it's in a prokaryotic cell, it would happen in the cytoplasm. I switched the when and the where. Oh my goodness. All right, so where and when, just switch those. My mistake. So it happens where is in the nucleus. When it happens is before cell division. So where, and then this right here is going to be when it happens. All right, why does this happen? Like I said, this happens as a part of cell division for reproduction. So genetic copies are made so that a cell can make more cells. So this is part of reproductive processes in cells. All right, so let's look at this process. It sounds like it would be very fancy and complex, but it's actually very simple. So the way that DNA replicates is the DNA molecule will actually unzip. So we have two sides of the DNA ladder. I'm just going to draw the DNA right here. Okay, so I'm going to start off with my DNA molecule. And here is the rungs of my ladder, those nitrogenous bases. And so the way this process works is the DNA is going to unzip. So the first step in the process is the DNA unzips, and that means it separates, it breaks apart. And when this happens, hydrogen bonds are broken that are holding together the nitrogenous bases. So they are broken, and the thing that breaks them is an enzyme, because enzymes break things apart. So they're broken by helicase. So helicase is an enzyme that is going to break the hydrogen bonds. And you guys might want to make a little note to yourself, like, who does this? It's helicase. All right. So what happens is, here comes helicase, works like a zipper, and it's going to go down and unzip the DNA strand, breaking apart those hydrogen bonds. The next thing that happens, step number 2, is free floating. So there's free floating nucleotides. So free floating nucleotides are going to bind to their complementary base. So for example, adenines will bind to thymines and cytosines will bond to guanines. So A's go with T's and C's go with G's. And these things are going to be put together by another enzyme, because enzymes put things together, called DNA polymerase. So it puts together the polymer DNA. So DNA polymerase is going to put together the new DNA strand. And so these free-floating bases, let me get a good color here. There we go. So free-floating bases are going to come in and they're going to pair up with their complementary base. And they're going to be putting together a second complementary strand. So they're going to all bind together. All right, the hydrogen bonds reform between the bases. So hydrogen bonds reform between the original and the newly forming strand. Okay, so hydrogen bonds reform between the bases. And in the end, we're going to end up with two identical DNA strands. And each will have half of the original strand and half of the newly formed strand. So we say that this is semi-conservative, meaning one strand remains the same and then we have a new one. So we end up with two identical DNA strands, each with half the original strand. So you can see here's the original strand. Now that's the original. And then here we have, the part that was newly formed. Okay? But in the end, they're going to separate completely and have two. All right. I hope you have a wonderful day.